So I have a really boring title for my talk, so I thought instead I would just show you this image instead of the title. So this is not an FA image. This is an image that's generated with tractography. So what I did is I just seeded streamlines from everywhere, and then we don't tend to represent single streamline in my lab. We do probabilistic tractography, so we construct histograms, voxel-wise histograms that tell us the probability that a streamline will end up in some part of the brain. So this is a voxel-wise histogram of streamlines tracked from everywhere in the brain. And as you can see, it shows that there's quite a lot of information in the white matter in this data. And because it's generated through tractography, and tractography is done in continuous space, these histograms can be binned at any resolution you want. So even though the data here was 2 millimeter isotropic, this image is 250 microns isotropic. And, some people call it track density. I think it's very pretty, and because it's kind of dynamic, you can sort of see the fibers running through the brain. Sadly, the reason I think you can see so much structure is because tractography is not good enough to pick up all the white matter. So there you go. My talk is quite short, so if you want, I can keep this on for a little while. We've got time. So as you heard from Christian Beaulieu this morning, tractography can be used to tractography can tell us where the white matter bundles are in the brain. It's very good at that, and there's quite a lot of known white matter tracks that we can easily reconstruct with this technique. And we can use these as for ROI placing to do quantitative analysis of these bundles. But you should always keep in mind that tractography is based on the assumption that diffusion is easier along the axons than, on the observation that diffusion is easier along the axons. And we consider the direction of easy diffusion as a proxy for axon orientation. So there you go. So we can we reconstruct these trajectories of least hindrance to diffusion, and we consider them proxies for connections. But you've heard this before. Obviously, this bridge between diffusion and tissue orientation or axon is not as straightforward as this throughout the white matter. You can have complex situations. So in this example, it's just some toy illustrations. When you have parallel fibers, all is fine. You can consider the tensors telling you about the orientation of these fibers. If you have fibers fanning within a voxel, or crossing, or kissing, or bending, tensor doesn't really tell you much about this information. And if you kind of believe that this orientation is the orientation of the underlying fibers, you might miss situations like this. And even if you consider more complex models like crossing fiber models, they can still not distinguish some of these cases. So because there is this because it's such an indirect measure of tissue configurations within the voxel, tractography makes mistakes. So for example, ambiguous situations such as these, which we can mistake for crossing fibers, in the white matter, our streamlines can jump between one tract and another. And near the gray matter, other things can happen. So for example, you may have some bias towards reaching some parts of the cortex instead of other parts. And if you want to build connectomes of the whole cortex, then you're going to start to see these biases, and they're going to bias your estimate of network connectivity and all the rest of your interpretations. So I call these kinds of errors modeling errors, which I distinguish from errors due to the measurement noise. And these modeling errors So although tractography is very good at localizing tracks, so telling us where the cingulum is, where the corticospinal tract is, etc., we can get, it's not as good at telling us where in the cortex we end up, where in gray matter, which it's not as good as telling us about site-to-site connectivity. And that's because diffusion to axon mapping can be ambiguous. We know the orientations of diffusion, but we don't know where within that voxel each orientation is happening. And of course, this might sound trivial, but we cannot quantify these modeling errors. So if we had an underlying truth, then we could tell how far away we are from that truth. But if we had that, then we would use that as our model. So obviously, we cannot tell how far away from the truth we are. But we can reduce this distance from the truth by improving modeling. And in my talk, I will mainly talk about how we can improve this situation by having better data. And this is just an example of what better data can give us. It's an early example that we've come up with while writing this, while people were writing this connectome grant, trying to demonstrate what we can gain from higher field strength, more directions, and better tractography algorithms. So in this example, we were looking for somatotopy of the corticospinal tract. So in blue, green, and red, you have we were looking for connections coming from 3 different places on the motor strip. And we were looking for different somatotopy in the thalamus and in the corticospinal tract. So this descending tracks kind of split before reaching this point and kind of feed the corticospinal tract and the thalamus separately. So if you try to do that with when we try to do that with the standard data that we used to see in Oxford back then you kind of sort of see this green versus red thing, but it's kind of blurry and you don't really distinguish thalamus from corticospinal tract. And we had access to some 7T data from Minnesota back then, and it worked beautifully. And these blobs were actually separated, so we could find not only somatotopy in the corticospinal tract but also in the thalamus, and they were distinguished from each other. And you can start to see that when you increase the number of directions from 60 to 256. So in this talk, I'm gonna present some of the data that's been some of the data, the diffusion data that's going to be acquired for the Human Connectome project. So I'm going to talk briefly about what the Human Connectome project is. I'm going to show some of the advances that have been made for making data better. I'm going to concentrate on the diffusion side of this data acquisition process. I'm going to talk a bit about preprocessing. Today we haven't heard much about preprocessing, but I think it's really important. So I'm going to talk about two aspects of those preprocessing. For this Human Connectome project, we've been particularly anal about preprocessing. So I'm going to show you that it can really make a difference to the quality of the data you get at the end. And I'm going to try to show you that the kind of avenues for new research in methodologies that can be open when the data quality improves. So you've all heard of the HCP. This is the website. This is the kind of current website where all the info is. So I invite you to all go there and read about it. There's now this new band here. It's the Connectome Workbench. It's the visualization software that comes with all the Connectome related softwares. It's really good at visualizing things on the cortical surface. So I really encourage you to play with it. It's quite cool. These are the Connectome players, or the main players. This is from an all-hands meeting that happened last year. David Van Essen and Camille Eugerbil are heading this project. And these are the Oxford people. So we're mainly involved in the analysis side of things, both in the DTI and the fMRI. So the goals of the HCPs primarily provide data to people, and using the new technologies and also new methodologies to make the data as good as possible, and also to come up with new strategies to define connectomes, so to parcellate the brain and to measure connections between brain regions. So 1,200 subjects will be scanned with functional and structural data, including resting states. What I mean here by functional is resting state fMRI. But there'll also be some genotyping, lots of behavioral testing, and task-based fMRI. And these tasks are kind of a fairly standard task designed to cover as much of the brain as possible in order to kind of confront those with the parcellation data. And one very important aspect of this project is that the data will be easy to access and interact with online, which is a major challenge because it's quite a lot of data. I think the strength of HCP compared to other data sharing projects, apart from lots of subjects, is that the data quality will be extremely high, and all subjects will be scanned on the same scanner. And also, the pre-processing is going to be very, very anal. And we just finished 2 years of optimization, so I can show you what the data will actually look like rather than what we think it will look like. So one thing that will be used extensively in this, both in the functional and in diffusion data, is slice acceleration. We call this multiband. So basically, the idea is to acquire more data by acquiring several slices at the same time. And the way it works is that you excite multiple slices simultaneously, in this case 4 at a time, so multiband 4 or MB4. And then use the fact that you have multiple coils to unalias your signal using the coil sensitivity profile. So each slice is seen more by some coils than others, and then you can use matrix inversion to recover each slice separately. So effectively, when you have an MB factor of 4, you can acquire almost four times the data in the same amount of time. So this is really, really good for both for fMRI and for diffusion for obvious reasons. So in the case of diffusion, we can use that to acquire more directions, for example. In fMRI, you can use that to lower your TR. So in diffusion, we're going to be using 3 times acceleration, so MB3. And I think in the fMRI, they'll be using MB6. So as I said, the advantage of slice acceleration is that we can acquire more data in the same amount of time. Also, the scanner is equipped with a high gradient, so it's a dedicated scanner, Skyra Siemens scanner, 3T scanner, and it has a 100 millitesla per meter gradient inset, and that means that we will have more SNR. Basically, we can get to higher B values at lower echo time. So what can we do with these two things? We can, with lots of SNR, we can go for higher resolution. We can acquire more orientations, or we can acquire more B values or shells. Because we're going to have a slot of 50 minutes of scanning, we decided to do all of these. This is what an early attempt at using these kinds of accelerations on diffusion data. This is what the raw data looked like. So this is an acquisition, a 3-shell protocol, B1000, 2000, and 3000. This is a 1.5 millimeter data, isotropic, and it's been using MB3. so this is 2 times 150 directions per shell, and the directions are intermingled, so effectively it's 450 directions. And 2 times, we acquired that 2 times for reasons I will explain in a short while. So the first thing we noticed when we saw these images is that even at B3000, we have loads of SNR left. So well, it looks even better on my screen here, but there's lots of signal left here. So we thought, why not go even higher resolution? This is 125 millimeter data at B1500. Still lots of signal left. So we decided to go with this resolution. And actually, the data looks fantastic, especially if you're interested in where is it in the cortex that you actually reach when you do tractography. The anatomical detail is much higher than I was used to. This is a little bit of an unfair comparison. I mean, it's the same subject, but it's 2.5 data. Most people now acquire 2 millimeter data. It's quite clear that you see many more details than here. So now I'm going to talk about two things two sort of tools that have been developed in the context of the HCP for preprocessing, and they've been developed by this guy Jesper. Basically, we wanted somebody really careful and anal to do this implementation, and even though he looks very jolly, he's actually put it in front of a computer. He becomes really anal, and and he's been tackling two problems. One is to do with the field inhomogeneity induced distortions, or EPI distortions, and one is to do with the specific kind of distortions that diffusion data have, which is EPI eddy current distortions. And here are two tools. One of them is going to be released quite soon, and this one slightly later, Topup and eddy. So for EPI distortions, we chose to go with the option of changing the phasing code direction. So acquire the data twice with reversal of the direction of traversal of k-space. And when you do that, the distortions change direction. So you can use those two images to calculate the warp, and then apply those warp to the data, and then combine those two data in an averaging type way to get the signal back. And yeah, this is how Topup works. So you acquire your two images, you combine them, you get the field, and from the field you get the warps, and then you get a corrected image. And it works really well. For the Connectom projects, we apply in left-right phasing code instead of anterior-posterior. So that's why the images look asymmetric. They look quite ugly here, especially here in the bottom. This is the other direction. This is the corrected one. So you can see that not only the anatomical detail, the anatomy is kind of recovered, but also the signal. I don't know if you can see, but for example, this bright thing here gets back to less bright. So this is only if you use a field map, for example, and only use one of the two images plus the field map, you can't recover signal. You can only recover geometry because it's an ill-posed problem. But if you combine those two things, you can recover the actual signal. So that's for Topup. The other pre-processing that Jesper tackled was to do with eddy currents. So in diffusion imaging, you apply these two large gradients before you acquire your data, and you assume that these gradients are kind of square. When you switch them off, they switch off immediately, but they actually don't, because when you switch them on and off, that causes eddy currents in the scanner, and those can interact with the readout phase. And and what that causes is that the diffusion data look like they're displaced or sheared, and they are displaced and sheared in different ways. So you're going to apply these gradients along different directions, so they're going to interact with positioning along different axes. So when you look through the images, they kind of jump around. And added to that is motion. So we need to deal with these two things at the same time. And what people generally do in order to correct for eddy current distortions is that they take a B0 image, which doesn't have these eddy current distortions, and then they register. We do that too. Up until now, we register all the diffusion weighted images to this to a B0 image. The problem with this approach is that it's not very accurate, mainly because the contrast in these images is different from this one, and it's actually different for each diffusion weighted image, since you apply gradients in different directions. And when you go at higher B values, it's even more of a problem. There isn't actually a match. You can't really match these to the B0. And so Jesper came up with a different solution, which is kind of a model-based solution. So he's using something he calls a Gaussian process prediction maker. So all this red box does is it predicts what the data should look like. So it's a model of the data. And this model works in kind of brain space, so undistorted space. And then you can use the kind of susceptibility-induced distortions, so the EPI distortions, and a model of what the eddy currents are along the eddy current distortions are along for each of the gradients. And then using this predictive model, you can predict what the scanner data look like, so including both types of distortions, and compare to the actual data. Then you can do an optimization on this aspect of this whole loop. It's kind of iterate a couple of times between prediction and optimization of the eddy current model, and that works really well. And because it's trying to register, it's trying to register two images that have the same contrast. It doesn't suffer from this problem of registering to B0s. And basically, when you look at the images as a movie loop, they don't jump anymore. So it works really, really well. And it even works for, we've tried this on B values up to 10,000. Yeah, it's quite a robust piece of code. The only problem with this is that it's very, very slow. So what did I write here? So it uses Gaussian processes. It can include this susceptibility-induced field calculated from Topup. It has different models of these eddy current fields. So it has linear and quadratic model of the eddy current fields, and it works for high B value. So, so this kind of pre-processing, it does buy you voxel resolution, because obviously when data is registered very well across the different gradient orientations, then you effectively enhance your resolution. So this is why it's quite a shame to acquire data at 1.25 millimeter and then lose that just because the pre-processing is not careful enough. So now I'm going to show just a few examples of what you can do with high res. This is an example of the arc rate fascicle, where you compare to kind of the more conventional way of acquiring data. You see more of these nice branching through the cortex, which is what we're after. This is exactly the things that we are after for the connectome. So we're not only concerned about where the major bundles are. We're concerned about where they end up in the cortex. This is an example trying to separate the external versus extreme capsule. Actually, this is the HCP data. This is post-mortem data that Carla Miller and Jennifer McNabb, should have written their names here, but acquired in Oxford. And this is quite high-res data, so it works really beautifully on this particular brain, and it's starting to work for the 1.25 data. One last example, which is trying to see more of these branchings in the white matter. This is tracking from the primary motor cortex in the hand area and trying to see if those projections kind of if they have different splittings through their trajectory. And we noticed that there were like 3 blobs here near the thalamus, and these ended up at different parts in the brain. One went to the all the way down to the spinal cord, one to the brainstem, and this one here in the thalamus. And we couldn't see this kind of details on the 1.5 millimeter data. And this is a coronal view of the same experiment, where you can see these kinds of branching along the course of the corticospinal tract. And we think that these 2 branchings in the thalamus can also be seen in some of the tract tracing data in macaque monkeys.
Can I really quickly ask, what kind of tractography algorithms are these? I love the resolution and I love the images. Is that probabilistic?
Yeah, it's the probabilistic algorithm in FSL. So it's not based on the diffusion tensor model. It's based on the ball and stick model. So it's kind of a crossing fiber model, where each fiber is a kind of degenerate tensor that only has one eigenvalue. It's a very simple partial volume model. But obviously the data will be lots of the models that we've heard about today will be applicable to this data, including the high-D stuff, since we're going to be acquiring multiple B values in multiple directions. Actually, I didn't tell you about the final protocol, but it's here. So 1.25 isotropic, 3 shells, 1, 2, and 3,000 seconds per square millimeter, and because it's quite high res, we can only do 2 times 90 directions per shell. But as I said, these directions are intermingled, so it's effectively 270 directions, and each direction is acquired twice in order to do this top-up kind of correction. A total of 540 volumes. That's 50 minutes of scanning. So I think that this type of quality of the data can trigger quite a lot of new, well, hopefully will trigger quite a lot of new methods, ideas. And I'm gonna give 3 examples of the kinds of methods we've been working on in order to get more out of these data. I'm not going to show you applications to this actual data, because we kind of done these before the data was actually available. But we really hope that it's going to not only give us good connectomes, but also give us good methods. So the first example has to do with multiple B values. So I told you that the final protocol has 3 B values, but you might wonder why is it that we are acquiring 3 shells? Why not just acquire 1 shell, the best one of the 3, and then cover more directions for that shell? And the idea is when you look at diffusion signal, it looks like this. So this is just a representation of what the diffusion signal looks like in different directions. So imagine a circle. Imagine your signal is around a circle, and you apply diffusion gradients along different directions. And when you apply these gradients, you get signal attenuation. And you have more attenuation in some directions and less attenuation in some other directions. And the more attenuation you have, the more diffusion you have along that direction. So the key in detecting orientation in diffusion imaging is to find these directions where you have more attenuation. And obviously, when you increase the B value, so going here from 100 to 2500, when you increase your B value, you increase your contrast between the different directions. And this helps you find those orientations, those fiber orientations. Obviously, in this connectome project, we are not interested, the only tissue property that we are interested in and we want to get right is orientation. We're not interested that much in kind of fiber density or other tissue properties. We're interested in orientation. So we want to get it right. But the problem is, because it's attenuation that gives you orientation, you have to fight against SNR. And the idea is that high B value will give you contrast, low B value will give you signal, and maybe combining the different B values can give you a good compromise. And this is another example showing, this is simulations showing the advantage of B values versus, for different types of SNR. And this table inside table shows our ability to reconstruct crossing fibers we've simulated crossing fibers. So when you, the more white you are, the more crossing fibers you've actually detected. So it's better to be white than black in this particular example. And this is the number of directions, and this is the SNR. And within each box, you have B value and the kind of angle between fibers. This is because we know that it's kind of intuitive that when you're 90-degree crossing, it's easier to detect than when you're close to each other. And what you can see here is when you increase the B value, you kind of increase your ability to detect fibers that are closer and closer to each other because of this contrast idea. But you need SNR for that. So we thought by looking at this plot that we could kind of combine kind of squares from different parts of this table in order to get the best estimate of orientation. And the problem when you start changing the B value is that you start moving away from some of the assumptions that we make when we fit models to data. And one of the assumptions is that diffusion is mono-exponential, but experimental evidence shows that it isn't when you start to even when you just this non-mono-exponential decay starts at a B value of 1,000. And this is not accounted for by most models that are fit to the data before tractography. So there are many reasons why you may observe this non-mono-exponential decay. Some of them are interesting from a tissue perspective. Some are less interesting, like partial volume effects. We don't really care too much which one of these things is causing this non-mono-exponential decay, but we want to account for it. And the reason we want to account for it is because if you don't, then you start overfitting fiber orientations to your data. And when you start overfitting fiber orientations to your data, you start overfitting streamlines and finding connections where there are no connections. And the reason why you overfit is explained in this example. So here I just generated data and plotted them in the same way as I showed you before, using kind of multiple diffusivities into the same model. And when you try to fit a mono-exponential model to this data, and this is multi-shell data, so you have 1, 2, 3, 4, 5 shells here, and you try to fit all of them at the same time, then some of them are fit well and some of them aren't. So you have more signal attenuation in the inner shells than you have in the actual data. And that's the best you can achieve with one fiber model. But when you add more and more fibers, what happens is these fibers that are orthogonal to the main fiber orientation, they have more attenuation in this direction, but they have less along this direction. So they kind of elevate the signal of the inner shells, and they start to try to mold the shape of these inner shells by adding more and more crossing fibers. And these are the ones that we want to avoid. Oh, really? Okay. So I'm going to skip this slide. We just came up with this gamma distribution model that allows us to model this kind of non-exponential decay. And this is real data. So in the corpus callosum, you expect one fiber orientation, and it shows that it doesn't overfit. It fits one fiber in both. So in the top versus the bottom, if you try to fit a one fiber versus a three fiber model, in this case, the gamma distribution model only allows you to fit one fiber and kills the other two fibers. And this shows a whole brain picture of that. So in red, yellow, and blue are voxels where 2 or 3 fibers were detected with the mono-exponential model, and these are mostly in the interface between tissue and CSF, and we think it's the actual cause of this non-mono-exponential decay is partial voluming. The other methodological advance that may be possible with this high resolution, high SNR data is modeling fanning of fibers within a voxel. And this work's done by Stam, who's sitting just there. So this is how you write Stam in Greek, apparently. So when you look at the whole white matter, it's fiber bundles are not necessarily all going the same direction along the whole course. They can fan out. And when you zoom in within a voxel, you might find some trace of that fanning even within a single voxel. And typically, we model crossing fibers, we model single orientations, but we don't model fanning. So Stam came up with an explicit model of fiber dispersion. So this is his ball and rackets model versus the ball and stick. You can see that if you just use the ball and stick, you may mistake fiber dispersion for a crossing. You can get false negatives in your tractography. And this is data from macaque, very high SNR macaque data where we were able to fit this model robustly. We weren't able to fit it robustly to our standard data that we had at FIMRIB, but hopefully with this new connectome data, we can start to do that. And you can see fanning here near the cortex. So hopefully these biases towards the top of the gyri will be compensated for by this fanning near the cortex. And the last idea I wanted to talk about, it's actually a shame I don't have enough time because it's the coolest idea in this talk. It's a new idea we call rubiks, which is to do with which stands for resolutions unified with Bayesian inference and crossing fibers. So lots of people talk about combining B values, either to fit interesting tissue properties or to get good estimates of orientation. But nobody really talks about combining voxel resolutions. And in the world of diffusion, it actually makes sense to do that. Small voxels will give you good precision or accuracy about where fibers are and where the orientation of fibers is spatially, but they will have low SNR. And big voxels will give you lots of signal and lots of ability to kind of robustly infer orientations, but they won't tell you where things are. And we thought, why not acquire data at both resolutions and combine both data sets into the same model and then try to infer parameters at the highest resolution? And furthermore, what you can do is, because these voxels are big, they have lots of SNR, you can actually use a different B value for the two resolutions. So use as high B value as you can for the big voxels to get lots of contrast to noise ratio, and then be quite gentle with the low resolution, but still be able to kind of separate things spatially. So Stam came up with this model which basically combines two data sets, but infers parameters at the high resolution. And it improves things. This is simulations, but it actually also improves things on real data. So in these simulations, the one on the left uses only high-res data, and the one on the right uses both low and high-res data. But crucially, these two data sets are matched for acquisition time. So, so when you have the same acquisition times, it may be worth kind of stop acquiring your high-res data and then start acquire low-res data, because even if you acquire more high-res data, you won't get more. You can get more if you acquire more low-res data. And this slide is a bit complicated, but it shows that what rubiks is doing is not just smoothing. It doesn't smooth, it's actually even though it's combining a big voxel with the small voxels that is inside it, it keeps kind of the resolution of those small voxels. And that's because this red line from rubiks is in the center. And when you smooth the data, this red line goes to the left. What this histogram shows, I can't explain it in detail now, but what it does is it compares the orientations within voxels that have been combined in the model to voxels that haven't been combined in the model. So you would expect, if rubiks was smoothing, you would expect these to look much more similar to each other than these, and they actually don't. But they do if you start. But they do if you start doing an actual smoothing process. So that's the end of my talk. I just have one last joke, which is that my title was boring, and I saw the program, and I was jealous of Christian's title because he had this cool KISS idea: keep it simple, stupid. But I thought that maybe I could have chosen this title, the kickass principle in diffusion imaging, and the take-home message is with better data come better modeling opportunities, hopefully. These are the people who were involved in this optimization phase for the HCP from Oxford, University of Minnesota, WashU, and UCB. And if you don't want to read their names, you can look at this instead. That's it.
We still have time for questions and comments.
A quick question. Where does the Bayesian come in, the Rubik procedure? Because you didn't mention that at all.
So, we have a generative model. We like to work with generative models, which means we have a bunch of parameters that generate data. And in this case, they generate both high-res and low-res data. And the low-res aspect of it. So imagine you have a big voxel and you have smaller voxels inside it. So the generative model for the high-res data is kind of the standard, either the ball and stick model, and for the bigger voxels, it's a partial volume model. So it was sum of the high-res voxels. So that's a generative model. And whenever you have a generative model, you can and if you make assumptions about noise, then you can use Bayesian inference to kind of invert the model.
Since you've mentioned twice, I believe, in the talk that you want to make data available to public, what are the terms and conditions under which it would be released?
This is I obviously don't know the answer to this question. You need to ask David Van Essen or read terms and conditions in the website. I'm sure they're there. But I believe that NIH wanted to kind of release the data, make it easy to use, and also people don't have to, for example, include co-authors from the HCP in their papers if they use the data. They just need to kind of acknowledge. So I think it's this type of terms, but I'm not 100% certain of any of the above.
Okay, so you mean that it will not be as disastrous as eddy, right? Where you need to put eddy, or where you need
No,
to put all the
it's definitely
consortium?
not required to have
Okay.
co-authorship from this.
Okay. And could they produce maybe banners from and sell them? Would be their non-commercial restriction and all those? Okay, but I'll ask later.
Yeah.
Thank you.
Hi, Saad. I have some technical questions. First is the field, the geometric inhomogeneous correction. You acquire two-phase encoding direction. That is for each diffusion weighting direction or just for B0 image?
So you do that for each of your images, all the volumes, but you only use the B0 images to calculate the field. So these are high SNR images, and you can acquire a bunch of them, not only just two. And you can use those to calculate the field and calculate the transformations, and then apply this transformation to all your images.
So that actually the scan time wouldn't increase that much because you only do that on a B0 image.
Oh, okay, I
Right?
see what you mean. So, there are two possibilities. If you don't have enough scan time, then what you can do is, and you still want to do geometric distortion correction. So you can acquire all your data using just one blip direction. And then for your B0s, acquire a few B0s with the reverse blip. So that doesn't have that much scan time cost. The cost would be that you can't really recover signal when you have so when these distortions, what they do is that they can blur signal across many voxels or squash them into the same voxel. And if you want to kind of unsquash, it's an ill-posed problem. And it stops being ill-posed if you have both stretching and squashing. Then you can kind of use both informations to recover the signal. But if you don't have, if you only care about geometry in these areas, then you can do what you suggest, which is acquire just one blip direction and then correct for geometry.
Sorry, I have a second question is about your eddy current correction. So my little experience is I feel that Philips scanner is smaller eddy current artifact than the OG scanner. And also the new G scanner, like a G750, has less eddy current artifact. Do you have maybe other people has?
Do you have any serious questions? I don't know much about GE versus Philips versus Siemens eddy currents. Maybe Stam knows.
Well, I don't know for this particular comparison, but it's also a 100 millitesla per meter gradient. So the distortions are larger than we normally see.
The multiband sequence, which we got very excited about when we saw the first article, but it's really only been implemented on Siemens scanners. And I saw some people involved in that project at a meeting in March, and they said that they have a grant to port it to other platforms like Philips. Do you know anything about that? Are you?
No,
don't,
I don't. You need to ask Camille and those kinds of guys. Okay. I know that they're quite happy to kind of share the sequence with others, but I don't know about porting it to another type of scanner. But they've already shared it with a few labs that have Siemens scanner.
But you have to have a Siemens scanner.
I think. I don't know.
Well, the current program is only for the Siemens. Yeah.
Okay, so I wanted to ask you about the B values.
Yeah.
Sorry. Yes. So I want to ask you about the B values, and you had 1,000, 2,000, and 3,000. It's on. Yeah. And so how did you come up with that? I mean, what is the intuition? I mean, I would guess it would be linear in Q instead of B value, and that would be my guess.
So, so probably the reason why you said linear versus.
So I mean, if I had 1,000 and 3,000, the one in the middle I would be middle in Q value, so probably a bit closer to 1,500.
So our approach to answering these kinds of questions was kind of data-driven rather than theory-driven. So there were lots of factors to optimize, not only the b-values. There's even the shape of the pulses were kind of investigated and optimized. So kind of multi-dimensional optimization problem. And and our approach was always to kind of try different options, look at the data, fit some models to the data, and do some quantification of these things, and come up with a measure that actually quantifies the accuracy of estimating fiber orientation, because that's the only thing we cared about. And all these numbers come from this kind of optimization process. But as for the b-value, going too high in b-value didn't benefit us. So above 3,000–4,000, data quality went too low, especially at this high resolution. So we decided to stay within that range, and within that range we tried different combinations of b-values. And it didn't make that much difference which combination we choose. It made a subtle difference, but we still took the one that we thought was the best, and that was how the numbers came up. So it was purely data-driven rather than theory-driven. We didn't want to make assumptions about, the ADC profile, or we didn't want to make those kinds of modeling assumptions.
So there's a bias towards you want orientation. That's the only thing you want.
That's right. Yeah.
So, thank you.
So, can you tell us a bit about the 1,200 subjects? I know I could go read this on the website, but you're a captive website spokesperson, so. So
they're very nice people, from every single one of them. So the criterion there is they want to have families with twins, okay, and non-twin siblings, so they can do all sorts of genetic calculations. And they're going to be from the kind of Midwestern area.
Yeah, there's a famous Minnesota twin study, which
yes,
would be, because the scanners at CMR are, right?
No, it's at WashU.
WashU, oh, okay. I
so
guess
it's the Missouri Twins project.
Oh, it's the Missouri Twins study, okay, thanks. So is there going to be any age distribution, or are you just going to have to take what you get in the twins?
It's 21 to 35, so
okay,
it's young adult.
Okay, thanks.
I have a question. So you do most of your tractography, I assume, in FSL, right?
Yes.
Do you have any, like, in terms of, there's lots of parameters in there in terms of like step length and number of steps for the tractography. Do you have any like strategies as to, when I do this stuff in my data, I tend to just use the default, but I don't know why. Like, do you have any strategies based on your voxel resolution and everything for how you should choose those different parameters?
So the two you mentioned, step length and number of steps, it's just the choice should be driven by your voxel size, and intuitively a step length smaller than your voxel would be my recommendation, and the number of steps depends on whether you're looking at small brains like macaque brains or something, or big brains. 1 meter is not a reasonable distance, so something that's that can go from one side of the brain to the other should be enough. Yes. What other parameters are you concerned about?
Well, I mean, there's just a few to choose from, and I just tend to use the default because I don't know any better. But if there was something more optimal, I was just curious.
Yeah, I use the defaults myself most of the time.
Thanks.
Quick question. There are additional artifacts from cardiovascular pulsations and all that. Are you recording physiology, or are you triggering by cardiac pulse, or do you do anything about that?
We don't do triggering, but there was a plan for doing physiological monitoring, but I'm not quite sure if that plan is still on or not. Is it?
Good. And what is the plan to release the data? When it becomes available? When do you scan? What's the plan?
Steve?
The first quad was actually collected this last weekend. And the goal is to release quarterly. However many subjects were scanned in the first 3 months will be released at 3 months. But it will probably not start till that rollout probably won't start till the end of the year. So it'll be like 5 months instead of 3 before the first ones come out, because things, not surprisingly, are still a little bit in flux, even as we're collecting the first real data after our optimization phase. But it is meant to be made primarily available to anybody that wants to use it, and there's a release agreement that's pretty gentle. I mean, you don't have to sign away your children or anything.
Or can I give it to someone else?
I'm not sure what the final decision is on that, but for some of the pilot data, it was if you give it to somebody else, you're supposed to let, there's 2 sort of levels. If you give it to somebody else, I think it's going to be if you give it to somebody else, you have to, or you're supposed to let us know that you're passing it on to otherwise invisible people to the project. But I don't think there's any, it's very, you know, it's complicated depending on what it is you're passing. And there's a lot of palaver about that in the agreement. But it's pretty, I think you'll find that it's pretty gentle. I mean, this is meant to be the country's data. That's a big part of, or the world's data.
Well, there are multiple sides of the coin here, right? There is ethical and IRB approval and all those, right?
So that's why there is some control on it, because you're not going to get access. You're not going to get access to identifying data.
Exactly, but then there is no, theoretically, there should be no restrictions on whom I give that data to.
But so you're right, it is complicated, because in some cases the kind of analysis that you want to do will require you knowing the twin status, for instance, of the people. In that case, you're going to have to sign a more stringent agreement that you're not going to pass along, and that you're going to keep the data, since these quads are odd, right? So it's 2 twins, and you'll know whether they're dizygotic or monozygotic. You'll know their age, and you'll know the sexes of all 4 of the people. That is highly constraining, and so it's identifying information. If you're going to do an analysis that entails having that information, you're going to have to sign a much more stringent agreement than just having access to the data. You know, give me all your resting state data on all the males from 20 to 30.
Okay.
So that kind of data will be very easy to get and very freely given. The more information that you want about the subjects, the more stringent sort of agreement that you're going to have to sign.
Sounds very fair, and the only comment would be I could recommend maybe existing licenses where you could make that data publicly available under existing license. Actually, license is even not correct term, because data itself in the States is not even copyrightable. So it's the collection to which you could kind of grant.
So let me tell you, I haven't been involved in the process, but this has been done with the advice and oversight of a large number of people, including people who've been doing this sort of thing in the past.
Yeah, but we have experience with those people who had it in the past, and now.
I think it's. I'll reiterate. The goal is to have the first order information very freely available.
Okay. Thank you.
And the more information that you want, obviously, the more stringent it's going to be.
And what about the genetic data then?
So the genetic data is being collected and stored, and there is going to be no analysis on it for some period of time, with the hopes that we will be able to do very complete description of the genetic analysis. Right now we don't have enough money to do everything that we would want on all the subjects, but it's like electronics, the price of this is coming down each month, and so not a lot of analysis of the genetic data is going to be done until later in the collection, and then that will be made available. But there's a lot of sort of general population genetic information by virtue of the fact of the design itself. So you're not going to get molecular genetics, but you'll, you know, depending on how much information you get, there'll be a lot of sort of heritability appropriate information available quite quickly if you're willing to sign the more confidential agreements.
That would make sense that with the genetic data, that then if you start to have information plus some of the data and plus genetic data, you can see how you can narrow down. You know, if I know part of your genetic and you know that on top of that, you are a homozygote twin, you know, then it starts to become relatively easy to pin down. Yeah. No, on top of that, if you have brain information, then you may end up having even unwittingly more information than you realize that you have, and that can create the obvious problem that therefore I think that the agreement that one would need to sign should absolutely take that problem into account.
Well, so like the very generic data, you'll say, I want, you know, one of each quad, for instance, so that you aren't being tainted by the fact that you unwittingly have twins in your groups. So the idea of this is why the informatics side of this is very complex. What information that you want for the kind of question that you want to ask is going to be very different. So if you just want to do the biggest sort of population study that you can for resting or diffusion with this really high quality data, you're probably not going to want to have twins involved in that. So you're going to end up with, hopefully, 300 people who are unrelated except, you know, in the sort of statistical sense. So that kind of data will be really, you know, free reign. But as soon as you start getting into the more genetically informed kind of design, then of course you're going to have potentially identifying information. But the whole idea of the informatics infrastructure is allow you to query the data set to get this sort of set up these sorts of questions.